Let's take a look at our smooth muscle. So we're back in our muscle chapter here, and I'm gonna scroll down, let's see, to our, let's see, we're gonna do smooth muscle. So let's kind of scroll down here. And you might have a section of just smooth muscle that is pretty, just kind of a longitudinal section. And then a lot of you are jumping to an actual like organ or larger structure like an artery. And those are completely acceptable as well. You just need to make sure that you have your atlas out and turn to an artery or turn to the small intestine, a particular section of the small intestine. So you have a little bit of a guide because it looks very different if you have just like a section of smooth muscle and then you're trying to find it in an actual organ. Okay, so this one's pretty easy, and then this can be a little bit more complicated. So if we just look at a longitudinal section of smooth muscle, it will look something along this line, right? And if we increase magnification, you can see that there is kind of like a lot of texture going on here. So from this border pretty much up to here is a longitudinal view of smooth muscle cells. And from here to there is looking at the cells in a cross-sectional view. And this is very similar to what we would see in the lower part of the esophagus. We would see actually an additional third layer in the stomach, but then we would see this very same pattern as we get to the rest of the intestinal section. Okay, and so you have to kind of get your kind of bearings of what is longitudinal versus cross-sectional view. When you start to kind of see this wavy pattern that you see here with lots and lots of nuclei, it kind of resembles dense regular collagenous connective tissue, quite honestly, but there are differences between that one and this one. You might also see some of these breaks in the tissue, which can be a mixture of blood vessels and also nerve fibers that are traveling through, which we'll take a look at a little bit later in the term. But again, look for this kind of wave-like pattern that you see, and that's very, very indicative of smooth muscle. And so I'm not so concerned with cross-section at this point, but if we go in with a higher magnification, you can see that the nuclei, again, are very dark staining. Each nuclei represents one individual smooth muscle cell. I talk about how these cells are spindle shape, and you might say, like, I don't see that here. I just see a lot of this pink background and there's no membrane. I can't see that. And you would be right. So we know that the cells are spindle shape, and we teach that they're spindle shape, but you might get here and go, I don't see that. And so the membranes aren't kind of being stained for that, right? So what we would assume is that the center of the cell is where the nucleus is located. So this is the thicker part of the cell, and then the cell essentially tapers out to the side. And then you have one cell stacked on another, and they're kind of like irregular shaped or spindle shaped cells or tiles that kind of stack together. So I'm going to pull up a diagram here and kind of show you the basic appearance of a smooth muscle cell. So I headed over to one of the medical websites and pulled up some lovely diagrams. And so here is our typical spindle-shaped cell, right? And we also use the word fiber, which again is very confusing, but it's a living cell. And we have a very elongated nuclei. You can see that the thicker part of the cell is around the nucleus, and then it has these tapering ends. And they kind of stack together, right? Like tile would, and they're closely bound to each other. And we don't see striations in these cells because the protein on the inside is arranged in kind of a cobwebbing fashion. So it's not stacked and arranged in such a way that we see striations like we see in skeletal and cardiac muscle. The protein fibers are there. They're just, again, more in a cobweb. So when they contract, the whole cell kind of scrunchels down, and it actually can kink the nucleus even in the inside. It kind of has like a inchworm kind of appearance sometimes. So it might be elongated like this, or maybe kind of a little bit crunched a little. And so they're showing you more of a relaxed versus a contracted state. If I scroll up here real quick, you can also see that they're outlining for you in this photograph the actual spindle-shaped cell. And again, when you're looking, you can't really see the borders all that well. Here you even can see them better than the slide I'm showing you before, but they're outlining that so you can kind of get that general sense. If you look at this in cross section, then you can see they just look like a bunch of circles, right? You can see the nuclei, but then they're just all these kind of irregular shaped circles because either you're cutting it here or here or here, and then you get these kind of different diameters of the actual cells. And so you have the ability to see both of these sections on the slides in your slide box. So you have kind of a longitudinal cross section because they're two bands of cells. So bear with me. Let me jump back to the website and let me scroll out a little bit. So these are longitudinal, these are cross-sectional. So if I zoom in here, you would see the long version, and then you would see kind of the circular version, and that is essentially here. So longitudinal versus cross-sectional view. This site also will show us, that's cardiac, but they're showing you also kind of a side-by-side view. So I do tell students, if you have the time, to pull out 3 microscopes and set them side by side and put, put your skeletal muscle on one, and then put smooth, and then put cardiac, and then view the slides at the same magnification. So view them all at 4, then 10, then 40. And so then you get a nice sizing idea also of how big these cells are in comparison to each other. You can also see here that skeletal has striations, as does cardiac, but smooth does not. There's multiple cells per nuclei for skeletal, but cardiac and smooth only have 1 cell per nuclei. The cells here for for skeletal are long and gigantic and kind of tubular, and for smooth we know they're spindle shape, and as we'll see for cardiac they're a little bit more of this kind of branchy appearance, right? So some basic differences kind of right off the bat visually from what we can see. So if we jump back to our slide here, again, all of these purple staining nuclei are representing each individual spindle shape smooth muscle cell. And then you may have the ability to see some of the connective tissue that does help to support these cells as well. And you can find blood vessels that are here too. Again, nerve fibers, we haven't got into all of that yet, but that is there. So if you're thinking like, what is this other tissue? This doesn't look uniform. We'll know that there are other things kind of cutting into that tissue. So this is a nice, cross-section longitudinal view. You have a slide like this one, but let's go ahead and head back out to our muscle area. And even though this is mesentery, you do have an artery vein nerve slide that would be very similar to what we see here. And so let's just go to the giant artery right away. And we can see by looking at this in cross section, you can see that there is 3 layers to the vessel, which we'll talk about in another video, but you have the intima, the media, and the adventitia, and this medial section here is all smooth muscle. And so this is a great slide to use. You can definitely use this one to show and represent smooth muscle. So the endothelial layer is here, and then lots of smooth muscle, and don't confuse that with the dense irregular connective tissue outer layer. And you hopefully won't, because they stain differently. There's a different color to them, and also there's a different texture. So this is there's a lot of nuclei in both, so that might kind of throw you off, and you're like, oh, is it the same? And note that no, it is not. And if we go in with closer magnification, again, you might see some elastic fiber being, stained in here because there's a lot of connective tissue in our vessel wall. But these are all of those nuclei for each individual smooth muscle cell. So some of them do have kind of a kinked appearance because the cells are kind of in a partial contracted state, and some of them do not. But again, lots of our cellular material. A little bit of this stringy stuff in here is going to be our connective tissue. Okay, so whether we pick a big artery like this one or this one over here, you could pick a vein, I suppose, but it's going to definitely be a little bit harder to make out your smooth muscle because there's not as much muscle here in our vein, and even that's not a good idea. So I think if you are, really skilled in finding some of this tissue, you'll be able to see it. This one just has so much more fiber, protein fiber kind of weaving into the smooth muscle. So I would really kind of stick with the artery guy over here and, kind of work your way in from the endothelial lining outward. Okay, so let's go back out and take maybe a look at an intestinal area. I'm going to say to stay away from the esophagus. You can, but the esophagus, I don't know where they took your esophagus section from. If it's in the upper esophagus, then the muscle that's in the wall of the esophagus will be skeletal. If it's towards the bottom, it will be smooth. If it's somewhere in the middle, it'll actually be a mix of skeletal and smooth. So that would be fun to try to figure out what you have, but if you're trying to learn it off the bat, I would say stick with like the smaller large intestine. So the intestine section that you have will be, if you're looking at small intestine, you'll have all of these kind of projected villi areas, and then there's just a lot going on. So what I want you to do is try to find the lumen where your food would be flowing through, and then head on out to the periphery. And so you're going to see these big bands of pink out here, and that's going to be primarily your smooth muscle. And so we're just going to kind of keep clicking in. And again, this is all villi and crypt material here. And then we really want to make sure that we're not hitting connective tissue, which is here, and we really want to get into muscle. You might be confused a little bit because it's like, well, that looks like this, and you would be right. So that is actually a little bit of smooth muscle right here, which we'll talk about when we get to the intestine. But that's what we call the muscularis mucosae, and that is a little bit of smooth muscle. And then you have a span of connective tissue here, and then you run back into smooth muscle again. So some of the connective tissue can actually throw you off because it does look so similar to smooth. That's why I say don't worry about that. Just come out way to the periphery, and you're going to see a section that typically when we cut this for your slides, it's going to look cross-sectional view. And then here is some longitudinal fibers. Your slides sometimes might be, I don't want to say it's reversed because that's not exactly what I mean, but it might be reversed or flipped, and this might be cross-section and this might be longitudinal because of how they cut your intestine. So if they cut your intestine longitudinally or if they cut it in cross-section, that affects these two layers and how they're seen, because this is called our inner circular layer. So it travels around the circle, around the actual circumference of your intestine, and this layer is outer longitudinal and it runs down the length of your intestine. So the cells are traveling with the length. So regardless, try to find longitudinal versus cross section, and again, it might be reversed on your slide. But on this one, we can clearly see that this is a longitudinal view. Okay, so we go ahead and we can click in and again see all those lovely nuclei, and then all the pink background is just representing our cells. We can see some blood vessels in there too. And then clearly this is all in cross section. Look at all those little circles, right? And again, we can see nuclei every so often, pretty indicative of smooth muscle. All right, so a couple different ways to essentially look at this particular tissue. All right, thanks guys.